Welcome everybody to this presentation called Quantitative in Silico Modeling of Diabetes Microphysiological System allows for translation to humans. And what I will talk about is how we take the type of data that can be generated from microphysiological system, MPS systems, of the same type that we heard in previous presentations, and then we can analyze these data using computational models and translate to humans in a way that's useful for um, uh, healthcare, for drug development, and just for inspiring people to lead a healthy life. And I will talk about how all of these are interconnected. And this MPS system, it's been developed in a collaboration between Tissues and AstraZeneca. And it has, at the moment, two compartments: a liver compartment where the liver cells are spheroids, which are developed either by donations or coming from cell lines, and a pancreatic compartment. And then there is a blood flow or a micropump in between. And we also have other versions which I won't talk about today, where we also have a heart. Here we have simulations, which are the lines. The areas here are the uncertainty of the simulations. And then we see here that even though this is predictions, so the model hasn't been trained on these data, but it's predicting these data, we can see that it's a good agreement between the data and the model. And it's that for the first meal response. And then you change the media, so you have new fresh media, and you see in the model, you can see this is what we believe the second meal response would be, and the third, and so on. And then you see a couple of things. You see that there is an impairment over time, so you have less and less glucose uptake, which is the same thing you see in type 2 diabetes. And you also have less and less insulin production. And you can also see in the model, you can see many things that we haven't had the time or the resources to measure. And you can also look at things that you haven't measured at all, like this beta cell functionality and so on. And apart from simulating or testing the model and using it to simulate things that we haven't measured, you can also translate to humans. So you can go from this long 48-hour meal response. So this is like a 2-week period here. So you can go from a 48-hour meal response to the normal 4-hour meal response that you see in humans, simply by doing things that we can see that we should do. So changing the organ proportions, for instance, so that you get this normal meal response. So we understand these differences and we can rescale in the computer. So finally, just some acknowledgements. So lots of funding from different sources, and in particular Tissues and AstraZeneca have developed the experimental data that we have done the modeling for. And the modeling has been done by Bill and Casas.